I'm titled my talk is Migraine, Microbiome, and Metabolism. And let's see how we forward this. So there's a well-known saying, a very old saying, that you are what you eat, right? So and this is getting a little more traction these days because it's becoming more and more clear that your diet and your gut microbiome can actually influence your behavior. And this is especially obvious from some of the studies that people have done in people with neurological disorders, because their microbiome seem to be quite different from healthy people. So there's this correlation between the microbiome and behavior and even neurological disorders. But the correlation is not materialized in really causality at this particular point. So how the microbiota and the microbiome can actually influence behavior is not entirely clear. And today I want to talk to you about the model system that we use and an approach that we use in order to really get at the fundamentals and the causality between microbiome and metabolism and behavior. And I'll divide my talk in three parts. The first one being migraine, the second microbiome, and the third metabolism, as the title already suggested. Maybe to some surprise that you can actually study this guy very well in a tiny little worm, the worm C. elegans. Right here, you can see C. elegans. Can you see my pointer as well, or my little hand here? Or should I use the laser?
Yes, we can see the pointer, it's fine.
Okay. So here you can see C. elegans. It's actually crawling around, it's laying eggs. You can see it's feeding on E. coli. That's the laboratory diet for C. elegans. It feeds on E. coli and it lays eggs and does all kinds of things that make life interesting. And C. elegans is actually very good to look at the role from microbiomes and behavior, because it has actually quite a simple nervous system. Here you can see C. elegans. C. elegans is about 1 millimeter long. This is the intestine. Here is the feeding organ. This is the head of the animal. The animal has about 302 neurons. Most of these neurons you will find in the head of the animal, which constitutes kind of a primitive brain. And you also find a lot of neurons along the ventral cord of the animal. And these are very important for worm locomotion. And just to, and we know a tremendous amount of detail about the nervous system. We know exactly how it's wired, how all these neurons are connected. You can do very easy genetics and the lifespan of C. elegans, so you can regenerate C. elegans in about 3 days. So you can do experiments genetically very quickly. So C. elegans is a great model system. And the first part is that I would like to answer is whether worms can actually get headaches, right? So, and a lot of what we do and what Diego alluded to is we are looking at the escape response of C. elegans. We've done several genetic screens, and one of these genetic screens, we identified a mutant, which is right here. It's called, we named it ZUF35. Here on the left, you see a wild type. This is our mutant. And if you look at this mutant, you can see that it's slightly hyperactive and makes a lot of reversals, whereas this wild type worm is just cruising along straight, right? So here you can see this, for instance, if you plot speed against time. So once in a while, a wild type will make these reversals, but these particular mutants that we isolated from this genetic screen makes lots of them, right? So about 70 or something per minute. Now, Yongxi, a graduate student in the lab, decided to characterize what is the mutation that has taken place in this particular mutant, this hyperactive, hyper-reversal mutant. And it turned out that she found that this ZUF35 mutation results in a mutation in a voltage-gated calcium channel. These voltage-gated calcium channels are important for synaptic transmission. And this particular mutation is in this transmembrane loop right here, and it results in glycine to arginine mutation. And this is in a highly conserved region of this voltage-gated calcium channel. So this region is actually conserved from worms to humans, and it's a very conserved glycine to arginine substitution. Now, these voltage-gated calcium channels, as I said, they're very important for synaptic transmission. These channels, the CAV2 channel, are located at the presynaptic site, and upon channel opening, depolarization of the neuron, calcium comes in, and it triggers the fusion of synaptic vesicles with the membrane and the release of neurotransmitter. Now, Diego, when he was in the lab, actually, he characterized what is the effect of this particular mutation on the channel, right? So, and what he discovered is that this is actually a gain of function mutation. So these channels open more readily, so at lower membrane potentials, and there's an increased current density, so more calcium is going through it, and the channel inactivates slightly more slowly. So this really is a hyperactive channel. And this is also very different from loss of function mutations in this channel. So UNC2 was actually originally characterized by Sidney Brenner. Loss of function mutations were defined. And these loss of function mutations, they're here denoted in blue that we identified in the suppressor screen. These worms are actually very sluggish and uncoordinated, hence the name UNC, right? So they barely move, and you can imagine if you don't have proper synaptic transmission, then these animals do not move very much. By the way, before I forget, if there are any questions during my talk, please stop and interrupt me. And the interesting part about these channels is that in humans, there are different neurological disorders which are associated with mutations in this channel. So the human ortholog of the worm channel is called CACNA1A. And there are several mutations that give you basically two different neurological disorders. One is ataxia, and which is basically an uncoordinated behavior, much like in the worm. And other mutations can also cause a familial form of migraine, familial hemiplegic migraine. And the nature of these mutations, some of them are not entirely clear, certainly for familial hemiplegic migraine. So what we did is now take these mutations that you normally find in people that get migraine and introduce them as a transgene into the worm. And then what we find is also if you take these migraine mutations that you see right here, and now make a transgenic worms that express these migraine forms of the channel, then they also have this hyperreversal phenotype. So the wild type barely reverses, or does that about 5 or 6 times, whereas the gain of function mutation and the ones with the human migraine mutation, they have a much increased number of reversals. Now, what happens in the nervous system of this worm, right? So this gain of function, this UNC2 channel is actually expressed in all neurons in C. elegans. And what we find is that if you do electrophysiology and you look at the neuromuscular junction, that we find that these worms with the gain of function mutation have an increased level of cholinergic synaptic transmission, right? So that you can see right here. And the one thing that we also find is that if you now look in for GABAergic transmission, that we actually find a reduction. And this came as some of a surprise to us, because the gain of function mutation is also expressed in GABAergic neurons. So you would maybe expect that you would find an increase in both inhibitory and excitatory signaling in the worm, but that's not what we find. And now with the worm, because the worm is transparent, you can do some beautiful experiments by looking at the synapses, at GABAergic and cholinergic synapses. And what we find, if you now label the cholinergic synapse, and for instance, you look somewhere here in the ventral cord, then you see in the wild type all these little clusters, these puncta along the ventral cord. And the level of fluorescence kind of indicates the level of the expression of these cholinergic receptors. And if you now look at these ONC2 gain of function mutants, so these calcium channel gain of function mutants, then you see actually an increase in the expression of these cholinergic receptors. So you get strengthening of cholinergic receptors. And we find actually the opposite if you look at GABA receptors. So now you label GABA receptors with a green fluorescent marker, then you see kind of a reduction in the level of GABA receptor expression that's quantified right here. And just for the sake of time, I'm going to give you kind of the bottom line of what we found. We've done a lot of genetic experiments to dissect in order to figure out why is it that these GABA receptors are actually reduced. And what we find is that increased calcium channel activity in cholinergic neurons leads to strengthening of cholinergic synapses. So you find more acetylcholine receptors, and these acetylcholine receptors, so the increase of sodium and calcium activity in the muscle will activate calcineurin, which is called TAC6 in the worm, and that kind of destabilizes GABA receptors at the synapse. So in this way, by having this gain of function of this voltage-gated calcium channel, you generate this imbalance in excitatory and inhibitory signaling in the worm. And this is also kind of in line with what people find with a lot of neurological disorders, actually. So also with migraine, but it's also found with autism, depression, that there are imbalances in the excitatory and inhibitory transmission in the nervous system, right? So with our worm model of this familial hemiplegic migraine, we kind of see that this is indeed the case, right? So, and we can also kind of now start to dissect the mechanisms why you get this imbalance. And it may even give you some indications why some treatments, for instance, GABA agonist treatments that are used for migraine, why they might be beneficial to relieve some of the symptoms of migraine. So this story was published in eLife last year, or in 2019. So I've kind of gone through this kind of briefly because I want to talk about one story which we haven't published yet, and that is the microbiome, right? And what I mentioned before is that indeed the microbiome has been implicated in all these kind of neurological disorders, but it's utterly complex, right? So the weight of bacteria in your intestine is bigger than the weight of your nervous system, of your brain. And there are thousands of different bacteria that are present in the intestine, right? So to figure out why a certain bacteria might be beneficial or bad for you for certain neurological symptoms, to figure out the mechanism, the causality, is actually extremely hard to do this in humans or in mammals. Right? Now, we thought like the worm is actually very good to figure this out, because we feed worms a single bacteria. So you can give them a single bacteria as a food source. They actually can colonize the intestine, and you can look at behavior. Right? So you have a very defined system to look at the effect of bacteria on the nervous system. And as I said, in C. elegans, in the lab, we actually grow it on E. coli. It's not a bacteria that they normally encounter in an environment. So C. elegans in the wild, you actually find it in the decaying organic matter, decaying plant materials, rotting apples. And the lab of Marianne Felix and the lab of Gary Rufkin, for instance, they have looked in these environments where C. elegans normally lives at the kind of bacteria that it would encounter, right? So, and there are very different lots of different species that actually are found in association with C. elegans in the wild. So, Bukyu, a postdoc in the lab, he wanted to use our migraine, our headache worms, right? So by feeding them different bacteria and to see whether we can suppress this seizure-like behavior of these worms. right? So what he wanted to do is just feed these worms a different diet for a day. So give them not E. coli, but a different diet, and then analyze the behavior and see whether we can reduce these hyperreversals. And here, this is kind of the survey that Wukyu did. Here you can see all these different strains that were tested. And what he found is that there are a number of bacteria that significantly reduce the number of reversals, right? So here's the E. coli strain. So this is the regular E. coli strain from the lab. And then these strains with about a little over 60 reversals. And then these strains bring it back to around 45 or something. And it's actually quite intriguing is that if you take different bacteria, right? So here we have 3 different bacterial strains, Gluconobacter, Bacillus, and Comomonas. They all reduce the seizure behavior. But now if you give them a mixed diet, so if you give them the 3 different bacteria as a diet, you can really reduce these number of seizures almost to normal wild type levels. And I find this quite striking. And it also points that there are actually independent mechanisms, probably that there are different dietary factors or different interactions that these bacteria have with C. elegans that contribute to the suppression of these reversals. And so we wanted to figure out what is it that how do these bacteria actually make these neurological disease states slightly better, right, in the worm? And from all the bacteria that Wukyu characterized, he found that all these bacteria here in blue actually produce vitamin B12. And E. coli, the regular lab diet, actually does not produce vitamin B12. So we wanted to figure out what is the effect of vitamin B12 in this. And now the beauty is, so we focused on Comomonas because we can also do some bacterial genetics in Comomonas. So Comomonas makes vitamin B12. And now if you take a genetic strain of Comomonas that lacks a biosynthetic enzyme that produces vitamin B12, then you can see that this beneficial effect is kind of abolished. And even more, if you now just add vitamin B12 to E. coli, then you also see the suppression effect, right? So, and this is all in this UNC2 gain of function background. So these migraine worms, you see this beneficial effect. You don't see much of a difference in the wild type in this particular assay. Now, these bacteria, just like in the human intestine, they can actually colonize the intestine and just be there as live bacteria, right? So E. coli, the general lab strain, after about a day, you have about 100 of these bacteria in the intestine. So here, this is an E. coli strain, for instance, which is labeled with GFP. You can see here in the intestine the green fluorescence of these bacteria. And Comomonas actually is slightly better than E. coli, or this is a log scale than E. coli in colonizing the intestine. And here, as a comparison, we also took a very pathogenic strain, which is known to be very infectious and overgrow the C. elegans intestine is even better at this. But Comomonas also kind of colonizes the intestine of C. elegans. And now you can look like how long does it take before you see the beneficial effect. And so if you put worms on E. coli plus B12, then it takes about a day or 20 hours before you see this suppression in the number of reversals. Now, if you take them off, so if you now grow them for a couple of generations on B12, and then you grow them for a day without B12, then this effect disappears again. So this tells you a little bit about the time frame in which the effect of B12 takes effect and when it disappears. Now, if you grow them on Comomonas, however, so now if you compare these here in E. coli, it disappears after a day. If you grow them on Comomonas, then actually this effect lasts much longer, right? So only after day 4, it's completely disappeared. And this is actually correlated with the number of bacteria that you find in the intestine, right? So now we've taken them off E. coli, and then after a couple of days, by now only eating E. coli, then you see this particular change, and you see that Comomonas lingers on, and after 4 days, has kind of been replaced by E. coli in the intestine. So it's really the gut colonization that really is sufficient in order to suppress these seizure behaviors in our migraine-like worms. Now, how does vitamin B12 affect neural function, right? So how does that work? And I told you that in these UNC2 gain-of-function worms, they are very hyperactive and they have increased cholinergic signaling in the nervous system. And this is something that you can quantify in C. elegans quite easily by using aldecarb, which is an acetylcholine esterase inhibitor. So if you put worms on aldecarb, so acetylcholine cannot be broken down, so you get an accumulation of acetylcholine at the synapse. And now what happens is that these worms paralyze over time because they hypercontract, all their muscles become hypercontracted because of the activation of the acetylcholine receptor. Now, if you look at these UNC2 gain-of-function mutants, they actually paralyze extremely quickly, right? So after about 40 minutes, then these animals are completely paralyzed, where most of the wild type are not paralyzed yet. Now, if we add vitamin B12 to the media, then you can see that you get this shift in the paralysis. So these animals paralyze much more slowly. So B12 reduces cholinergic transmission in these UNC2 gain-of-function. Is there a difference in the wild type? No, in the wild type, actually, B12 does not much an effect. So if you're in a healthy situation, you don't see much effect in this particular assay. But if you're challenged, if there's increased cholinergic signaling, then you see the effect of B12. And is it that we only see this in the UNC2 gain-of-function mutation, or do we also see effect in the wild type or other mutants? And oh yeah, and I should say first before I go on, is that if we measured the level of acetylcholine in animals with B12 or without B12, then we find that vitamin B12 kind of reduces the level of acetylcholine. And here, it's not only in the UNC2 gain-of-function mutant. This is an acetylcholine esterase mutant, or a double mutant for 2 acetylcholine esterases. And these animals, because there's too much cholinergic signaling, they barely move. So here you can see tracks of worms that have been moving around for half an hour. And then you can see these little tracks right here. This is the wild type. This is plus B12. This is now one of these mutants that have defects in the acetylcholine esterase gene. They can barely move. Now, if we add vitamin B12, you can see that their locomotion is very much increased. And the same is if we give them Comomonas, a B12 producing bacteria, then you see an increase in locomotion. Whereas if you take a Comomonas strain that cannot produce vitamin B12, you don't see this particular effect, right? So vitamin B12 also here in an independent model reduces cholinergic signaling. And now, do we also see an effect in the wild type, right? So so normally, right? So there's not much, or if a worm just crawls on an agar plate, then they move very gently. But if you now make them swim, so actually you give them some exercise, because they have to they start now swimming and they thresh much more. So there's much more, and for these body flexors, you need cholinergic signaling in order to make them happen. So these animals have much more cholinergic signaling. And now what happens over time, so this is the same video or a different video, but now sped up, that over time, these animals actually go into quiescence. And it's basically they have muscle cramps. And now if you give them vitamin B12, then wild type animals, they have less of these periods of quiescence, suggesting also that in the wild type, vitamin B12 in situations where you have excessive cholinergic signaling might actually reduce cholinergic signaling. And I remember the first time I talked to Diego about this, is that he was saying that in his Bahia soccer club or football club, that they would take vitamin B12 injections to reduce muscle cramps. I don't know whether this is a typical Argentinian way. Might explain why you won in 1978, but nevertheless, so there might be a similarity also in humans. So just to summarize this part, right? So the Comomonas diet produces vitamin B12, and somehow vitamin B12 reduces cholinergic transmission. And so how does this work, right? So how does vitamin B12 reduce cholinergic transmission? And this is kind of the last part of the talk, and this concerns metabolism. So this is vitamin B12 here on the left. It's this cofactor. It's actually, we can't produce it. Humans can't produce it. It's only synthesized by bacteria, and we obtain it from either gut bacteria or from animal food products that also get it actually from bacteria. So if you eat meat or cheese or milk, that contains vitamin B12, and that's how we get it. And B12 deficiencies, it's known that it actually has severe impacts on nervous system development and also on nervous system function. It's well known that also for psychiatric patients, they sometimes prescribe vitamin B12. This is a well-known supplement that's given for neurological disorders, although they have no real clue just how it actually benefits these patients. So how does vitamin B12 affect cholinergic transmission? Now, in the worm, you can figure it out, right? So vitamin B12 is actually a cofactor for just two different enzymes in humans. One is the methionine SAM cycle, which is important for methionine synthase. And the other one is the uneven chain fatty acid breakdown. Right, so this is a methylmalonyl-CoA mutase. These two enzymes are the ones that use vitamin B12 as a cofactor. And in C. elegans, these genes are very much conserved. So you can find direct ortholog for all these different enzymes that you find in humans or in mice or in mammals. You find the orthologs in C. elegans, and this has been characterized by a colleague of mine, Marion Walhout. So now you can start to figure out which of these two B12 dependent pathways is important to modulate cholinergic signaling. And you can do this in C. elegans very easily by using genetics. So what you can do is now take our own two gain of function mutants, right? So with B12, you see this beneficial effect. And now what you can do is also introduce a mutation in either this gene or here from the uneven fatty acid breakdown or from the methionine SAM cycle. And what we find, or what you found, is that if you have a mutation in the methionine SAM cycle, the positive effect of vitamin B12 is actually abolished. And even more so, you actually, if you completely knock out the methionine SAM cycle, you actually get a slight increase in the number of reversals, suggesting that this pathway is important to modulate cholinergic signaling. Now, where is this particular enzyme expressed? So where is the activity of the methionine SAM cycle? And the way that you can do this in C. elegans is very easy. You can just make a transgenic reporter and hook up the promoter to GFP and make transgenic worms, and then you can see that this methionine synthase is actually expressed in the intestine, and it's also expressed in the hypodermis, but it's actually not expressed in the nervous system. So it's in the intestine, hypodermis, but not in the nervous system. And actually, by only expressing it in the intestine or the hypodermis, you can actually rescue the defect again. So it's needed in the intestine or the hypodermis. But for instance, if you were to express it in muscle, you don't see this beneficial effect. So what we have now is another little piece of this puzzle. So vitamin B12 from Komomonos diet is required for the methionine SAM cycle, and that inhibits cholinergic transmission. So now what is the link here? So what is the link between the methionine SAM cycle and cholinergic transmission? Now, in mammals, the methionine SAM cycle is actually very closely linked to the choline oxidation pathway. And choline gets actually converted to betaine, and betaine can serve as a methyl donor in the methionine SAM cycle. But choline is also a precursor for the synthesis of acetylcholine. And that normally choline is actually, it's a very important dietary supplement as well. Choline gets transported through a choline transporter into the nervous system, and there it gets converted into acetylcholine. So this is one way to make acetylcholine. So Bukyu in the lab, what he was trying to figure out. So in humans, you actually have two enzymes, BHMT and methionine synthase, or in mammals. In invertebrates or in C. elegans, you have one enzyme. They're actually closely related. And Bukyu actually purified the C. elegans methionine transferase. And what he showed is that indeed betaine and homocysteine can both act as a methyl donor in the synthesis for methionine. I should say betaine and 5-methyl tetrahydrofluoride. So both of these can act as a methyl donor, both betaine and tetrahydrofolate. Now, again, here, the enzymes that break down choline to betaine, you can look, we identified these orthologs, CHH1 and ALH9. And now, if you look in C. elegans as well, and these on2 gain-of-function mutants at choline levels, and what we find is that vitamin B12 also decreases the level of free choline. Now, if you add exogenous choline, so here with vitamin B12, you find this reduction. Now, if you add exogenous choline, you kind of abolish the suppression effect that we see right here. So exogenous choline abolishes this effect. Now, and also, if you look at these enzymes, this choline dehydrogenase and this ALH9, they're actually both expressed in the same spot as the methionine synthase. They're both expressed in the intestine and the hypodermis. So they're all at the right spot. Choline does affect the phenotype that I was talking about. And indeed, if you now knock out these genes, so if you now take a mutant, or in this case, it's RNAi, then you also abolish this positive effect of vitamin B12. So with vitamin B12, you get the reduction. Now, if you knock out this particular enzyme, you no longer see the beneficial effect of this of vitamin B12, suggesting that this is indeed an important pathway that links choline oxidation pathway with the methionine SAM cycle. And now, how do we get from choline produced in the intestine and the hypodermis to the nervous system? In C. elegans, there's a choline transporter, and you can also, that's on the next slide. So if you now knock out this choline transporter, then also you abolish this effect, this beneficial effect of E. coli. So what we think is going on to put all these pieces together, right? So what we think is happening is that Komomonos produces vitamin B12. If you have vitamin B12 in your system, if you have sufficient vitamin B12, then choline can be used as a methyl donor in this particular reaction, right? So that reduces the level of free choline in your system. And if you reduce the level of free choline in your system, then there are certainly under situations where you have increased cholinergic signaling, exercise, or UNC2 gain of function mutants, or migraine, or other genetic manipulations, then there's not sufficient choline to keep up with the level of acetylcholine release. So by adding vitamin B12, you reduce the levels of free choline, leading to a reduction of cholinergic transmission. So what I think is beautiful about this is that you can use a worm to really dissect how bacteria and bacterial factors that they produce, to dissect how diet and the microbiome can change behavior. And we found is that vitamin B12 reduces excitatory transmission. That might also explain some of the beneficial effects of vitamin B12 for people that are suffering from neurological disorders. And that we have discovered is kind of this new crosstalk between the methionine SAM cycle and this dynamic crosstalk between the methionine SAM cycle and acetylcholine synthesis. And this is only one of the bacteria that I talked to you about today, right? So this is a B12 producing bacteria. We've also found that some of the bacillus strains are also beneficial in this particular assay. It turns out bacillus makes nitric oxide, which is a different component. And we also have a bunch of different bacteria where we don't know at this particular point what the beneficial factor is. And we're currently trying to characterize some of these pathways and mechanisms. And with that, I would like to thank people in my lab, current and former. Diego was there, which was a tremendous pleasure, and it's also great that we're still working together on some of the other projects, and I'm sure Diego has talked to you about it. Some people that in Toronto from Meijin's lab that did some of the electrophysiology at the NMJ. The UMass Medical School has a bunch of C. elegans and fly labs, so it's a wonderful community. And last but not least, also the NIH for funding. And with that, I'd be happy to take questions from everybody. Thank you for your attention.
Okay. Okay, thank you, Mark, for the talk, for this very nice talk. So, okay, so we are open to questions from the audience. Yes, Osvaldo?
Yes, thank you, Mark. Thank you for the nice talk. It's very nice to see that you can reproduce a certain aspect of migraine in the worm. For a long time, we have been working on the FAH1 animal with collaboration with also with the Holland people from
Yes.
from Van Mandenberger.
Yes, exactly. And I recognize
okay.
your name. Yes, of course.
Okay. Yeah. Now, what surprised me of your work is that what I remember, I mean, from the neuromuscular, I don't remember exactly what is the anatomy of the neuromuscular junction or the nerve-muscle interaction in the worm, but from the vertebrates, which I know, if you increase the amount of acetylcholine release, you normally get reduced amount of receptors. I mean, you have a sort of feedback compensatory mechanism, and the other way around too. So I'm really surprised that you can get so much acetylcholine receptors spread all over the muscle. That's why I ask you, what is the relation between nerve and muscle in the worm? Is a close relationship like in the mammals or in the frog, or is a sort of diffuse interaction? Because that will make probably answer my question in a way.
Yeah, so there's always this this counterbalance between homeostatic plasticity versus Hebbian plasticity, right? So where if there's increased activity, you can strengthen synapses maybe, but for instance, if you dump acetylcholine on the synapse, you can also get resorption of cholinergic receptors postsynaptically to maintain homeostasis. And both of these processes also seem to occur in C. elegans, right? So if you leave worms, if you increase cholinergic signaling after development, so for instance, if you put them on aldicarb, then you also find this reduction of cholinergic receptors. And this might be similar to what you are referring to. But here in this particular case, on2 gain of function, so this increased cholinergic synapse is there from development, right? So from throughout development, so it's always there. So we think, or the thing what we see here is that you get more of a Hebbian plasticity rather than a homeostatic plasticity. So that's why we find the increase. But you're right? So if you just in an established nervous system would all of a sudden increase cholinergic signaling, then at the postsynapse you might find the reduction. So this is you find also both things actually in C. elegans. Yeah.
Thank you.
Yeah.
Okay. Another question?
Yes, please.
Yes, very nice talk. It was very, very nice. I was wondering about the behavior, about the reversal of the seizure. Can you explain a little more the mechanisms that is involved in this reversal of this behavior? Because in the first part of the talk, you mentioned that amitriptyline also has reverse, and amitriptyline is antidepressant. So yeah. I was wondering if it's not possible that a reversal of the motivation of the worm for different environment can be influencing the behavior when you obtain the reversal. It's just a question of curiosity.
Yeah, so there's a lot of work that people have done on reversal behavior, and they figured out all the neurons. So they're actually locomotion command neurons or premotor interneurons that drive either motor neurons that initiate a wave of muscle contractions from the anterior to the posterior, or from the posterior to the interior, and that allows the switch from backward to forward. Yeah, so it's let me so that is something that we know. So wild type worms usually they tend to move forward, and once in a while they will make a reversal to change directions. But here there's actually disturbance in these premotor interneurons. Yeah, so there's too much cholinergic signaling in these locomotion command neurons, where they become kind of unstable and they keep switching back and forth. So it's more at the premotor neuron.
It's more specific.
Yeah, and
more...
yeah, the GABA agonist that you mentioned. So we haven't really looked at the reversal part at this particular point. We have tested some of that, but that's more that we find this reduction, this excitatory inhibitory imbalance that is generated by this particular mutation. And it's well known that this antidepressants, they sometimes also use it actually for people with migraine. So maybe by activating the GABA receptor, you now restore kind of this balance somewhat, right? And this is, yeah, and depression is also thought to be this EI imbalance, where some people
kind of,
well. So by this, you kind of boost your inhibitory signaling in your brain again, in order to restore kind of normal brain function. That's the idea. Yeah.
They are different targets of action.
Yeah. So we don't, the GABA part is not necessarily much, not as far as we know, much to do with the reversal itself. That's more higher up in interneurons, not at the neuromuscular junction. Yeah.
Thank you.
More questions?
You can also
Yeah.
ask in Spanish if you want, but
Yeah.
maybe we need a
Yeah,
translator.
we can translate.
I am curious because I know that C. elegans doesn't have mutations that promote them a propionic aciduria or a methylmalonic aciduria. But in this case, when you have a mutation in this enzyme, so even do you have, you don't have any deficiency in B12, do you have also the same picture, the same symptomatology, or perhaps this will be the defect promoting the nervous system affecting in these patients with homocystinuria?
So this is if you have the mutation in the other B12
in the enzyme that has B12 as a cofactor.
Yeah. So we haven't seen anything obvious in nervous system function when we mutate the other mutation. There are some synthetic lethality, so these worms are viable in C. elegans. And there are some compensatory shunt pathways, which are also highly conserved in humans, which then get employed in C. elegans. And there is some lethality associated with that as well. If you have double mutants, if you mutate the shunt pathway and then the other pathway, but that's work from Marion Walhout. But we haven't seen anything behavior. We haven't looked very carefully though. So it could be that there are other behavioral effects there too. But yeah, at this point, we don't know.
But it will be interesting to check this in the patients with homocystinuria.
To see, or what would you like to check, the B12 different or B12 different?
Yes, if you can reverse with B12 or no, I suppose that you can reverse with B12 because it is a mutation in the gene of the enzyme. But if you have any connection between the mutation and the central nervous system affection of these patients.
Yeah, or maybe based on some of the C. elegans work, you can activate the shunt pathway or just to get rid of the toxic effects. Maybe those things are possible. Yeah, no, those would be interesting. That would be interesting. Yeah.
Thank you.
Thank you.
I can remember if you mentioned the effect of B12 on the channel itself, on the channel properties, of the calcium channel, of the mutated calcium channel.
Yeah, no, we haven't looked at this as well. This is it's interesting, right? Because as far as I understand, there are also some beneficial effects for B12 on people with migraine. But I don't know whether this is any change, effect on the ion channel itself. No indication of that. But we haven't looked in that way because Diego actually did these assays and by expressing it in mammalian in hex cells. But we haven't done experiments where we added B12 or not. No, we haven't done those experiments.
Okay. Yeah. Thank you.
So,
may I ask a question?
Yeah, sure.
Of course.
Okay, I am. Thank you very much. I do not belong to Sun Society. I am a microbiologist mainly, and I would like to know if you have ever thought of forcing the synthesis of B12 in Comamonas, for example, or another compound in other bacteria to see if we have a even bigger reversion effect by genetic modification of the bacteria.
So for vitamin B12, there are a lot of enzymes in bacteria that you need to have in order to make B12, right? So there it's a little bit hard. So we haven't done that experiment. So we have done those experiments, for instance, with nitric oxide synthase, right? So Bacillus makes nitric oxide synthase, and E. coli doesn't have that, and it also has these beneficial effects and these effects on behavior. And now if you can express kind of these bacterial enzymes from Bacillus into E. coli, and now also you get this beneficial effect. So yeah, the beauty of this system is that you can do the genetics. Well, you're a microbiologist, so obviously you know, but for me, this is something new, that you can do the genetics both in the host organism as well as in the bacteria, right? So, and eventually we hope to get to this if we resolve completely novel compounds to see whether we can maybe produce this in other bacteria, maybe even probiotics and things like that, but that might help neuronal health. Yes.
Very nice. Thank you.
Yeah, thanks.
Okay, Mark, there is a question in the chat from Eugenia Goya. You know her?
Yes.
So,
yeah.
So hi Mark, very nice talk. Nice to see worms in sun. Do you see protection against seizure in the progeny of worms filled with comamonas?
Where do you see it in the progeny?
Yeah. Do you see protection against seizures in the progeny of worms filled with comamonas?
So transgenerate, like
sure. Transgender.
Yeah, we haven't looked at that. Yeah. That would be cool to look at, but yeah, it would be cool to look at. So the colonization of the gut really happens only kind of in later. Well, Uge knows very well because she has done a beautiful study in Maria do Ciru's lab. But yeah, the colonization only happens kind of later. So I wouldn't expect it, but it's a very interesting idea. Yeah, it's an interesting idea. And there are little level, yeah, there are tiny amounts of B12 also in the media. So if you have absolutely no B12, then you eventually die after a couple of generations. So there are some transgenerational effects of B12, for instance, but so it's an interesting idea. We've never looked at it. Yeah.
Okay. More questions? So Mark, so in your migraine model, so the problem or the defect is an imbalance in the cholinergic and GABAergic receptors in the expression. You can rescue that with vitamin B12, but have you looked at the levels of expression of cholinergic and GABAergic receptors in anxious gain of function after the addition of B12? So just to see if the imbalance. Oh, okay.
No, we have not. Yeah, so we think that it's at the level of acetylcholine synthesis.
Just that, okay.
I think Young-Shi in the lab has looked at some point, but didn't see any obvious differences in growing them plus B12 or without B12 in the level of receptor expression. Yeah, these are also avenues to follow, but we think it's really in the level of synthesis of acetylcholine. Yeah.
Okay. So more questions?
Yeah. Hi.
Hi.
I have just one question. I don't know. Maybe you mentioned it during the talk, but I was wondering because if I'm not wrong, I remember that the combination of different bacteria in the diet give you a better even result that when you use one bacteria itself. Do you know why? Do you know? Because you have a signal, so you know that vitamin B12 is the one that could be mediating the effect, but do you know what happened when you have this combination of bacteria in the diet that you see a bigger effect?
Yeah, so we know it for one other bacterium. So if you Bacillus makes nitric oxide, and Comomonas makes vitamin B12, and then we have another gluconobacter where we don't know it, but it might contribute a third factor, right? So, so if you give them the mixed diet, they have both nitric oxide and vitamin B12, and nitric oxide actually reduces reversals by interfering with the oxygen sensation circuit. This is something that we're working on, whereas vitamin B12 might do this, would do this through the reduction in cholinergic signaling, right? So it becomes then a compound effect. So what we think is that different bacteria make different factors that each can contribute to the reduction, in this case, of these seizure-like behaviors, right? One being nitric oxide, the other one B12, and that these are independent mechanisms.
Okay, thanks.
So that's why we think it's common. Yeah.
More questions?
Mark, sorry.
Marcela?
Okay, Mark, I was surprised because you showed that Pseudomonas was better than E. coli to reduce the seizures, but it is very toxic. I know that you are looking only 3 minutes of the assay, but do Pseudomonas produce more B12 or
No.
Which is the difference? I don't know. I don't know if you said that.
Yeah. Yeah, so it turns out that different bacteria have different capacities to colonize the intestine, right? So E. coli is actually not a very good colonizer of the C. elegans intestine. And Pseudomonas, because it's part of the infectious state, is extremely good, right? So these it's very good at colonizing the intestine, and actually that induces the death of the worm at some point. So, but Pseudomonas also makes B12, but we've also used some Pseudomonas strains that do not seem to make B12. I don't know exactly. Yeah, it's interesting. I don't know actually whether B12 contributes to their capacity to colonize the intestine. Yeah, but it's an essential factor for C. elegans as well, for normal function. Yeah, so it holds on to it better. Yeah. Yeah.
Another question? So there are some mutations, for instance, in the myasthenic syndromes in humans that reduce the activity of cholinergic receptors, in particular muscle cholinergic receptors. So do you think, I mean, and the mutations or the positions are very well conserved. So do you think that that would be a good idea just to try vitamin B12 or better? So the bacteria with deficiency in vitamin B12 just to see if they improves the behaviors in those worms. You know what I mean?
And what worms then, Diego? Sorry.
So if you just recapitulate some myasthenic syndrome mutations that are so in the worms, so probably they will be uncoordinated for sure.
Yeah.
Do you think that just if you don't see probably as much phenotype in E. coli if you compare with Comamonas, because Comamonas has more, you know what I mean?
Yeah.
So maybe there is.
Yeah, so I think this is a wide open field, and
Yeah.
that's an excellent suggestion, and some of it we're doing, right? So we use kind of this ONC2 as a sensitized genetic background because it has a very easily scoreable phenotype. We actually use computer worm trackers in order to quantify the behavior. And so there's a very quick readout. We're also now have another mutant, AGL19, which is another calcium channel, which has been involved in Timothy syndrome and autism. But in principle, you can take any disease that you want, right? So if there's a worm ortholog, then you can test different bacteria. Worms are an extremely good system. And Uke has done this, for instance, for Alzheimer's, right? So Alzheimer-like phenotypes. It's also beautiful studies. They find something with bacillus. I think there's a lot of possibilities to work on this in C. elegans, and maybe modeling human neurological disorders, and then look at human microbiota even. This is something that we're now trying to do by taking human microbiota either from probiotics or from human stool samples from healthy and diseased patient, and give them to worms and then see if they can establish or if they can colonize the intestine in C. elegans. Yeah.
Nice. Okay.
So
more
sorry,
questions?
I have another question. I'm
Okay,
Eugenia
yeah.
here. Sorry,
Yeah.
I cannot make I don't have a video, but hope you can hear me.
I can hear you.
Hi, Mark. Yeah, very nice talk. I just heard that you mentioned about the NO effect from Bacillus subtilis.
Yeah.
So in that case, so we know that Bacillus subtilis is able to form biofilm, but it needs a couple of days to do this in the worm. I was wondering if the effect you see is actually much bigger after they formed biofilm.
Yeah, so we have done this with very let's see whether we've done those experiments where we look at much later stages. Let me think.
So the question is more related because if it's NO related,
right?
subtilis is able to produce much more NO after biofilm formation. So I was wondering if you may actually see a bigger effect in the seizure protection if you let them feed for more time.
Yeah, that's a great point. But at some point it becomes a little hard for C. elegans once they form the biofilm, it's a little bit hard to digest for them as well, right? So
yes,
we haven't done those experiments. What we have done, bacillus is not a very good colonizer in our hands of the intestine. When
that's right. Yeah.
Start feed them E. coli again, so kind of the effect disappears more rapidly, as for instance with Comomonas. But it's a good suggestion. Yeah, so I'll ask WooQ maybe to look at them if they are fed more or older bacillus, whether there's a bigger effect. We have induced nitric oxide synthase in E. coli, but I don't know the exact levels.
And have you tried to do the NO knockout in the subtilis?
Yes.
I mean, there is an available mutant.
Yeah. So in the nitric oxide synthase knockout, then the suppression effect is gone. Yeah. So it really is nitric oxide. Yeah. And we also have added this nitric oxide precursor to the plates, and then you can see it too. Yeah.
Okay. And then the third question actually I have is, have you considered that a mix of the different bacteria are actually somehow themselves modifying between bacteria? I mean, they are interacting somehow in the gut and doing something else. So some molecules that they can release. When you use a mix of bacteria, it's always difficult to conclude that there's something happening at the direct level, or just themselves interacting between them and then modifying their own behavior.
Yeah, no, I think you're entirely right that this is indeed a possibility, since the bacteria also have the individual effect, right? So in the reduction and that these effects seem to be additive. We opted using Occam's razor kind of for like they're independent things, but you're right. We cannot exclude that one bacteria uses a metabolite from another in order to make it. That's an interesting idea. And have you encountered something like that? Or
I can't say no, but I can't say completely yes either so far. But it's complex when you use a mix of different bacteria.
So in this case, we would grow them up separately to density, spread them out, and then immediately.
Exactly.
Yeah.
Or maybe you can kill one and then see if the other still does the same thing.
Yeah, we've done killing experiments, but not in the mixing experiments. But that's a good suggestion.
Okay.
Yeah.
Thank you. Very nice.
Yeah.
Okay, there is another question in the chat. Miriam, Is the ALK9 enzyme you mentioned orthologous to some of the human ALDH family? If so, do toxic aldehydes play a role somewhere in the B12 biosynthetic pathway, provided the enzyme is absent?
So ALH9 is a direct ortholog of the enzyme in humans that does the last step in the choline oxidation to betaine. So that was the first part of the question. Can you repeat the second part?
Yeah. Do those toxic aldehydes play a role somewhere in the B12 biosynthetic pathway, provided that the enzyme is absent?
Don't know where to completely comprehend it, because the B12 synthesis that's completely in the bacteria. But the B12 effect is completely gone when we take out this ALH9, so this aldehyde dehydrogenase. But I'm not sure whether that exactly answers the question.
Okay. Yeah.
Yeah.
Thanks. Yeah.
Right. Great.
So, any other question? Okay. So now those students or people that want to stay for a bit more, just talking with Mark about opportunities abroad or yes, living abroad or any topic you want. So you can keep